Okay, welcome everybody. This is the lab test review podcast for neurons, brain, and spinal cord. This will be on the last practical of the term. We again open with the cautionary message: do not use these PowerPoint images as a substitute for going to biosis or attending lab. This is simply meant to enhance your ability to do well on the lab practical. And what we want to start out with is a look at what we call action potential. Okay, your interactive physiology CD-ROM will have this information as well as the video content that I've posted on the lecture side of the discussion on the nervous system. So be sure to watch those videos and take notes on how an action potential is generated, how it propagates, and how it terminates. So in terms of action potential, you want to be able to describe when the different gates open and close and what each of these events represents. Remember that sodium and potassium are the main ions moving across the membrane that control gradient and action potentials. And you want to know what type of movement represents depolarization, which ion is responsible, what kind represents hyperpolarization, and the difference between a graded potential and an action potential. You want to be able to explain how action potentials are propagated along axons, both as a saltatory conduction and as a standard conduction method. And how does this influence conduction velocity? Remember from lecture that saltatory conduction is much more rapid than conduction across every square centimeter of the axon, which is again the process coming off the nerve cell body that carries the electrical signal away from the soma. Okay, so this is just showing you a sort of a quick summary here of how this operates. Briefly, what you want to remember about an action potential is that it's basically the sum of graded potentials at the axon hillock that reaches the critical trigger voltage. And remember that it's about minus 55 millivolts that allows those sodium gates to pop open. And when they do, sodium rapidly rushes into the cell, and this causes the resting membrane potential to go from minus 70 all the way up to about plus 30. And at that point, a couple of things happen. Number one, at that voltage, the sodium gates will shut and potassium gates will open, and potassium will rush into the cell and force sodium out. Okay, remember that there are leak channels that are operating the entire time in this process as well. So when that occurs, as the potassium rushes in, it'll actually rush in to the point where the membrane will hyperpolarize briefly, and then with the help of the sodium potassium ATPase and returning all the voltage gated channels to their original state, basically closed, then the pump can restore, along with the leak channels, the resting membrane potential of minus 70 millivolts. Okay, so remember that it's a series of gates opening and closing at different voltages that allow this to happen. Remember that a graded potential is different from an action potential. A graded potential fades with distance. Graded potentials can be excitatory or inhibitory, whereas action potentials are always excitatory. They're unidirectional and they're of the same magnitude. And once you start an action potential, you basically can't stop it. Okay, so what we're looking at here is just a curve showing you the different parts of an action potential, and we're asking which arrow is indicating a region of hyperpolarization, and the answer here is arrow 3. Okay, notice in arrow 3 that what's going on is we're actually below the resting membrane potential of minus 70 millivolts. This is due to the influx of potassium into the membrane as those gates remain open and shut very slowly, and the result here is that we actually dip below this resting membrane potential. This phenomenon is responsible for what we call the absolute refractory period, where there's no way for an action potential to go in the opposite direction, to back up, um, because of the nature of the membrane at that point not being able to respond to any electrical signal regardless of how strong it is. Okay. Next question is asking which arrow is indicating the region of repolarization, and that answer is 2. Okay. Notice that we depolarize here and we repolarize here. Okay. Coming back down. Next question, which arrow is indicating the region of depolarization, and that's arrow number 1. That's represented, characterized by the opening of the sodium gates, the rushing in of the ions through the channels, and the movement of the membrane potential from -70 all the way up to +30. Okay. Okay. A few more questions on action potential. Which arrow is pointing to the portion of the action potential when the sodium channels are open? And the answer to that question is number 1. Okay. The triggering event here again is reaching that critical voltage, about minus 50 millivolts, which causes those gates to fly open. And once that happens, sodium rushes into the cell. We depolarize all the way up to about plus 30 millivolts. And at that point, new events begin to happen. Next question, which arrow is pointing to the portion of the action potential when the potassium channels are opening? And the answer to that is number 2. Okay, what's happening here? The potassium channels open up. The potassium rushes out of the cell, and now the membrane potential begins to drop. And which arrow is pointing to a portion of where the action potential, where the sodium channels are closing? And that is number 3. Okay, sodium channels close, and the potassium channels remain open, and down goes the action potential all the way past the point where we have resting membrane potential, which is indicated by number 4, which is, the arrow pointing to a portion of the action potential when the potassium channels are closing. Okay, and you can see here potassium channels finally close after we've actually hyperpolarized, and now the sodium potassium ATPase along with the leak channels restore the membrane potential back to resting minus 70 millivolts, and we're ready now for the next action potential to come. Okay, so make sure and review those videos on the video in the video folder in the lessons tab on Angel on the lecture side of the course, and it'll detail each of these events and when and where they happen. Okay. Okay. Next question. At the point indicated by arrow number 1, is the majority of sodium moving across the plasma membrane in or out of the neuron? And the answer is the majority of the sodium indicated by arrow number 1 here is moving in. Okay. The sodium gates are open. The sodium is rushing in. Question number 2. At what point indicated by arrow number 2 is the majority of potassium moving across the plasma membrane, moving in or out of the neuron? And the answer here, number 2, the majority of the potassium here is moving out of the neuron. This is what is bringing the resting membrane potential back down, or the membrane potential rather back down, actually past threshold and past resting membrane potential, all the way to the point where we hyperpolarize. Okay. All right. Next question. Between axons A and B, which would conduct potentials with the greater velocity? Okay, and the answer here is B, because of the greater diameter. Okay, you can think of axon A as providing more resistance to flow because of its physical dimensions. B, you've got a greater opportunity to open and close gates. You've got more gates total, and as a result, the speed with which the electrical signal will move is going to be significantly enhanced. Next question. Of all 3 axons, which axon would conduct action potentials with the greatest velocity? And the answer here is C, because C not only has the widest diameter, but it's also myelinated. Okay, you can see the Schwann cells and the Schwann cell nuclei wrapped around the axon, and the result here is that the action potential will move very rapidly underneath this insulated part of the axon, and when it reaches the inner nodes, okay, these nodes of Ravnier, what will happen is that sufficient number of sodium gates are present in this region of the membrane that they'll pop open, and the action potential will regenerate and slide now to the next node, and as a result the speeds go up significantly with this kind of arrangement. Okay, a little bit on nervous system anatomy. The cell at the tip of the pointer is a neuron. How can you tell? Well, it's got processes that lead away from the soma. This is the soma, and these are dendrites. And if you look real close here, this long process here indicates that it's probably an axon. All right, so they're going to look sort of almost star-shaped. The multiple processes are there because the dendrites are part of what receives the electrical signal from other neurons that surround it. So they're kind of like antennae coming off the cell, and as these antennae are bombarded with chemical signals that either promote or inhibit the potential for this neuron to fire, all of those signals are going to be summed at this area called the axon hillock, and if we have sufficient graded potential to reach minus 55 millivolts, then we will fire. Okay, the action potential will propagate all the way down to the end of the axon, and the result will be the release of neurotransmitter at the synaptic bouton and the propagation of the signal to the next target. Okay, so be able to recognize those on a slide. We looked at those in lab over the last couple of weeks. Okay, next question. This is just showing you what type of tissue is shown. This is a chart. Okay, A is showing you myelin sheath, particularly that's a node of Ravnier right there. Okay, this is a longitudinal section through a nerve, and so what you see are many neurons, their axons, lying on top of each other, separated by connective tissue and by the myelin sheath, and it's these nodes that allow, again, the rapid conduction of the nerve signal from point A to point B. This is in all likelihood a motor neuron. Okay, node of Ravnier shown here at A. Okay, myelin sheath shown here in B and also in A. Okay, you can see the myelin, the white material here in longitudinal section and in transverse section. Okay, and then finally the axons are indicated by C. Okay, they are of course the processes that extend out from the soma. They are surrounded by connective tissue and by myelin, and the result is that the electrical signal can conduct more rapidly from the axon hillock to all the way down to the synaptic bouton. Okay, what portion of the brain is seen in this slide? This is cerebral cortex. Okay, and the two layers that you're looking at here, cortex here, and below this is subcortical white matter. Okay, staining sort of almost purplish pink. Remember that white matter is made up primarily of myelinated axons. That's why it looks white. So it's involved with signal transmission, whereas gray matter is made up primarily of nerve cell bodies, and as a result it's more involved with signal processing. Remember that the youngest part of the brain is actually the part on the surface. The neurons climb up the glia, and when they get to the surface of the brain they stop, differentiate, and they aid in the business of the brain, which is sending and receiving electrical signals and generating electrical signals. Okay, here we have a look at the nerve model. Okay, and you can see here a synaptic bouton. This is the structure that stores vesicles filled with neurotransmitter, which upon receiving the action potential from the axon will release the neurotransmitter, it'll hit the dendrite, and then depending on whether it's an inhibitory or an excitatory stimulus, um, you'll either prompt the membrane to move more towards firing or away from firing. Okay, all depends on the nature of the neurotransmitter released by this particular bouton. This is a dendrite. Okay, this is the axon. This is the axon hillock. This is the soma. Okay, this purple stuff are Nissl bodies. This is the nucleus. Okay, over here, what we see is the neural lemma. Okay, the axoplasm, which is the cytoplasm inside the axon, and of course the nodes of Ravnier, which are there primarily to speed the conduction of the nerve impulse from the hillock all the way to the axon terminal. Okay. This is a transverse section, and what you're looking at here, this is a Schwann cell, and what it does is it wraps itself around the axon. This is the axoplasm. This is the myelin. Okay, and this is the membrane of the Schwann cell in between these layers of myelin. And this is what helps insulate the electrical signal and speed its rate of conduction. Okay, so axoplasm, Schwann cell, and of course the myelin in between. Okay, moving on to the brain. We want to give the general name for this type of ridge. The ridges are the gyri, okay, and the grooves here, for instance, are the sulci. So don't confuse the two, gyri and sulci, okay. And there are some reasonably important gyri that we'll talk about, the pre and post central gyri, for instance, and some pretty important sulci that will address, the central sulcus being one of the major ones, and the longitudinal fissure being another one. Okay, we want to identify each of these labeled structures. Okay, this is the cerebral cortex. This is the corpus callosum. The corpus callosum is a set of commissural tracts that run between the right and left cerebral hemisphere and allow the right half and the left half of the brain to communicate with each other. And this is important because we have to coordinate what happens on the right and left side of the body in order to do things like perform purposeful movement. Okay, when this part of the brain is damaged, we sometimes have unusual phenomena that take place, such as phantom hand syndrome, where the right and left half of the body will operate independently of each other and often at cross purposes. This is the cerebellum. The cerebellum is important as a coordinator of sensory activity and motor intent. In other words, its job is to make certain that what we're trying to do matches up with what we're actually doing. Okay, so it receives input from the cerebral cortex, from the brainstem, from ascending pathways, proprioception, from cranial nerves, such as the 8th cranial nerve, the vestibulocochlear. All of this in order to make certain that what we're doing matches up with what we intend to do. This is the medulla oblongata, the pons, and of course the midbrain. And of these structures, which comprise the brainstem, and those would be the midbrain, the pons, and the medulla oblongata. Okay. Moving over here, more labeled structures. You want to be able to pick out the pituitary gland. Remember, it has two lobes, an anterior and a posterior. The anterior lobe releases trophic hormones that control the rest of the endocrine system. The posterior lobe releases oxytocin and vasopressin and is actually an outgrowth of the brain. The septum pellucidum is a thin membrane that separates the lateral ventricles from essentially the longitudinal fissure. So if we looked behind here, we would find the lateral ventricles. There's one in each hemisphere of the brain. There's a total of 4 ventricles in the brain, 2 laterals, a third, and a fourth ventricle. This is the thalamus, which is the gateway to the cerebral cortex and is surrounded by the third ventricle. The hypothalamus, which controls the activity of the pituitary gland among many other functions, and it lies just beneath the thalamus, hence its name, and the medulla oblongata, shown just below the pons down here. Okay. The ventricle behind the membrane at the tip of the blue arrow is the lateral ventricle. Um, the It's obscured from this view because the septum pellucidum is obscuring it. The third ventricle is posterior in anatomical position to the pons, or I'm sorry, posterior in anatomical position um, to the lateral ventricles. It's also posterior to the hypothalamus and the pituitary gland. It looks almost like a crow's head if we were to form a cast of it and look at the ventricles of the brain. Okay, indicated cavities here. You can see the third ventricle. Okay, the cerebral aqueduct, which connects the third to the fourth ventricle, which lies anterior to the cerebellum and posterior to the pons. And the job of the ventricles basically is to create cerebrospinal fluid and allow it to circulate inside and outside the brain, as well as inside and outside the spinal cord. And what does the cerebrospinal fluid do? It maintains the temperature and the pH and the electrolyte conditions around the neurons and provides a homeostatic environment for them. It's also a way to discard wastes from the activity, the metabolic activity of the neurons, and it's very important that there is always free flow of this fluid through the ventricles. If not, what happens is that pressure will build up upstream of the block, and if this happens, for instance, in the cranial cavity, what will occur is that the brain will begin to swell. It will press against the flat bones of the skull, and the result will be brain damage and perhaps death. Okay. If this happens in infants, this is called hydrocephaly. It results in an expanded brain case. We can fix this by putting a shunt into the ventricle that allows the CSF to drain into the abdominal pelvic cavity. This is something that has to be left in for the rest of the individual's life, but it allows the brain to go back to its normal dimensions and allows the cranial vault to shrink to the point where we'll now have the typical brain size and brain case size. Okay, the name for the pattern of gray matter seen here, this is the arbor vitae. This is the white matter that's inside the gray matter of the cerebellum. Remember that the cerebellum is the brain's sensory and motor coordinator, among other functions. The lobes of the brain are indicated here, the frontal, the parietal, the occipital, and the temporal. The significance of these? Well, we can touch on a few aspects. The occipital lobe is the area of the primary visual cortex. The temporal lobe, the primary auditory cortex. The frontal lobe is where we do most of our higher reasoning, and it's also part of what we call the motor association area. This is where we form plans that are going to eventually result in movement. We then send those plans to the precentral gyrus, and from there they head down to the brainstem, to the ventral spinal cord, and then out the spinal nerves to their target, and we carry out our activity. Parietal lobe is the home of what we call the sensory association area. This is where we assign a particular category to a stimulus, and it does that after it receives the information from the postcentral gyrus, which is the somatosensory cortex. And this is how the brain is able to interpret the stimulus that it encounters. Here you can see the lateral cerebral sulcus here. It separates the temporal lobe from the parietal and frontal. And you can see the transverse cerebral fissure that separates the cerebellum from the occipital and the temporal lobe. Okay, there's the temporal, there's the occipital lobe of the brain. The fissure that separates this hemisphere from the other hemispheres in the cerebrum is called the longitudinal fissure. Okay, the longitudinal fissure is connected. The right and the left hemispheres of the brain are connected across the longitudinal fissure by the corpus callosum, among other structures. Okay. Down here you can see the floor of the brain. You can see the olfactory bulb here. This is the first cranial nerve, provides our sense of smell. Here you can see the pons and the medulla oblongata and the associated cranial nerves. Here is the optic chiasm. Okay, this is where cranial nerve number 2, the optic nerve, crosses from right to left and left to right. This provides our sense of vision. Down here you can see the trochlear nerve, which is responsible for eye movement. Okay, it's a little bit difficult to see here. Remember that these two sides of the brain, this is the superficial side, this is the deeper side, and the trochlear nerve would be right about here. Okay, remember that there are 3 nerves that move the eyeball: the trochlear nerve, the abducens nerve, and the oculomotor nerve. Here you can see the optic nerve, the spherical structure that has been removed on this model and visible in the sheep brain is the pituitary gland. Okay, and remember that the pituitary gland is essentially the boss of the endocrine system, and its boss is the hypothalamus, okay, which lies just above it in anatomical position. Okay, here you can see the pineal gland, which secretes melatonin that controls our sleep-wake cycles. This, incidentally, is the choroid plexus of the third ventricle. This is one of the regions where cerebrospinal fluid is created by filtering the blood. This is the superior colliculus that receives and relays information having to do with vision, and this is the inferior colliculus that receives and relays information having to do with hearing, okay, as well as balance. And the collective name for these structures is the corpora quadrigemina, which is Latin for 4 bodies. Are these structures part of the forebrain, the midbrain, or the hindbrain? They're part of the midbrain. Okay. Remember that on the sheep brain we were able to see these structures by pulling down on the cerebellum, and you could see it in between the crevice of the occipital lobe and the cerebellum. Right back here, it looked like 2 pair of gluteus maximi. Right? We were joking about that class. Okay, labeled structure shown here. You can see this is a dissected sheep brain. This is the longitudinal fissure, the parietal lobe down here, the transverse cerebral fissure. This, of course, is the cerebellum. Okay, the occipital lobe is on the very back end of the brain here. Okay, this is a transverse section, and what you're looking at here are the cerebellum. You can see the arbor vitae in here. There is the corpus callosum, the commissural tract that connects the right and left hemispheres, the septum pellucidum, behind which is the lateral ventricle, the optic chiasm down here. This is where the optic nerves cross, and the thalamus. And you might ask, okay, well, wait a minute, where is the pituitary gland? And the answer to that is often the pituitary gland is pulled off when these brains are removed from the skull due to the fact that it sits inside the sella turcica of the sphenoid bone, which has a pretty tight grip on the organ. As a result, sometimes it is severed. Okay, into what cavity is this pin stuck? And that is stuck into the lateral ventricle. That pin goes through the septum pellucidum into the lateral ventricle of the right hemisphere. Okay, next question. Into what cavity is this pin stuck? And it is stuck into the fourth ventricle. Okay, the fourth ventricle receives cerebrospinal fluid from the third ventricle, which is right about here, via the cerebral aqueduct, which allows it to flow down, collect in this chamber, and then exit through median and lateral apertures to circulate around the brain. More structures to identify. We can see here the pineal gland, the pons, and the corpora quadrigemina. Okay. You can also see here the cerebellum, the arbor vitae, and here if we look at the dissected sheep brain, we have now pulled the cerebellum down, and we can see here the inferior and superior colliculus. There's the pineal gland. There's the transverse cerebral fissure, okay, and you can also see kind of into the third ventricle in there if you look really closely. Okay, this is the olfactory bulb. This down here is the mammillary body. This is a pair of protrusions that lie just behind the optic chiasm and slightly anterior to the pituitary gland, and they are part of the brainstem. Olfactory bulb, remember, is the first cranial nerve, allows us our sense of smell. Here you can see a cross section through the spinal cord. What we want to note here, this is the central canal. This is the dorsal root ganglion. This is the ventral root. This is the dorsal root. Okay, and right here, this is the dorsal furunculus. Okay, this ridge of white matter right here, and this carries ascending and descending pathways to and from the brainstem, between the brainstem and the spinal cord. Okay, moving on. More structures here. This is the dorsal ramus. Okay, this is going to branch out and head to innervate structures that are on the posterior body wall. Okay, this is the dorsal horn of the gray matter. This is the ventral horn of the gray matter. Remember, cell bodies are primarily in this region, and you can see here the ventral roots and the dorsal roots. Okay, remember the dorsal roots receive sensory information from the external environment, relay it to the central nervous system. The ventral roots transport motor information away from the central nervous system and to their targets. Here you can see the dorsal root ganglion, and you can see here, this is actually mislabeled. This is part of the ventral root, okay, and this is part of the dorsal root here, okay. It's covered by dura mater, all right. These are spinal nerves, okay. They're going to pass through the intervertebral foramina and out into their targets. Okay, so moving on. Dura mater shown here in blue. This is the arachnoid, this little layer of white, the subarachnoid space below it, and all the way down inside here is the pia mater. Okay, so we want to be able to distinguish between the three sets of membranes that surround the spinal cord: the dura, the pia, the arachnoid. Okay, just like in the brain. This is a cross section of the spinal cord in microsection. This has been stained. This is the gray matter. Okay, this is the ventral horn. This is the dorsal horn. This is the central canal, the lateral horn. This is the anterior white column. Okay, and these, of course, are the fununculi. Okay, that brings us to the end of the chapter on the nerves and the spinal cord. Remember, if you have any questions, make sure to contact me, and I'll be putting up a sort of mini practical for you, so that we can go over some of these structures in a Q&A format to give you some practice before we actually take the practical next week. Okay? Thank you very much.